The overall goal of this procedure is to identify genes within a population of individuals showing a preponderance of rare, functional variation. This is accomplished by first pooling a population of DNA samples. The second step is to create and sequence a next-generation sequencing library. This is followed by the alignment of reads to the reference sequence and the creation of an error model. The final step is computational analysis using the SPLINTER algorithm. Ultimately, SPLINTER analysis of pooled next-generation sequencing is used to show genes within populations harboring a preponderance of rare, functional variants.
Demonstrating the procedure today will be Francesco Vallania, who's a graduate student in the lab of my mentor and our collaborator Rob Mitra, and he'll be joined by Enrique Ramos, a graduate student in my laboratory.
The main advantage of this technique over existing methods such as single individual genotypers is that it allows you to detect very precisely rare sequence variants in a mixed population of DNA molecules without requiring any prior information.
This method can help answer key questions in the genetics and genomics fields, such as how to determine the frequency of novel disease-causing rare variants in large cohort studies.
Every SPLINTER experiment requires the presence of a negative and positive control to obtain optimal accuracy. Prepare the PCR reaction mix using PFU ultra-high fidelity DNA polymerase. The negative control is a PCR product from any DNA sequence known to be without genetic variation, such as a cloned vector backbone. Here, a 1934 base pair amplicon from the M13 MP18 vector is used.